Hello everyone, welcome to ITLS Academy. This is your mentor Dr. Shamaita Basu, and in this session we will be starting with a new topic, that is vaccines. We will be covering part 1 of this topic in this session. Okay. So before starting with what are vaccines, I would just like to give you a brief idea about the major historical milestones regarding vaccines. Okay, so this is basically the timeline which is evolving since the 1800s to the 2000s, that is the current time. So the first vaccine for the first time that was invented was the smallpox vaccine, as most of us might know already, in the year 1798. Then we have in the year 1813 Congress creates the National Vaccine Agency. Then in the year 1855 the first US law mandating vaccination for school kids was introduced. Then in the year 1886 the typhoid vaccine was introduced. In 1885 rabies vaccine was introduced. In 1896 we had cholera vaccine. In 1891 we had glycerin, which was used to reduce germs in vaccines. Then in 1926 or 1927 we had pertussis and tuberculosis vaccine, that is TB vaccine. In 1923 we had diphtheria vaccine. In 1924 we had tetanus vaccine. So consecutively 2 years we had two new vaccines. Then in the year 1935 we had yellow fever vaccine. Yellow fever is something which is now eradicated from the world. In 1936 we had influenza vaccine. In 1955 we had polio vaccine. In 1970 we had anthrax vaccine against anthrax bacillus. Then we have in 1963 we had measles vaccine. Then in 1977 we had pneumonal vaccine or pneumonia vaccine. In 1974 we had vaccine against meningitis. In 1981 and 1986 consecutively we had the Hep B and the Hep A vaccine. So first we had the Hep B vaccine and then we had the Hep A vaccine in 1986. In 1995 we had varicella zoster vaccine, and in 2006 we had HPV and the shingles vaccine, and in 1999 we had the rotavirus vaccine. Okay. Now coming to how these vaccines actually work. Okay. Okay, so basically these vaccines, as you can see in this slide, they help us to develop immunity by imitating an infection and triggering the antibodies to develop. So basically vaccines will be those parts of the infective pathogens or infective microorganisms which will be responsible for causing the disease. So they will just go inside the body, they will mimic that original pathogen, and the body will think that the body has been attacked by the pathogen, so it's an emergency condition. So the body will start producing or eliciting an immune response which will lead to production of the antibodies. Those antibodies will be available to fight the next time when the patient is exposed to that same virus or bacteria. So originally when the patient will actually be exposed to the disease or to the disease causing pathogen, then the body will already be ready with the antibodies to combat that particular disease, and hence the person will no longer suffer from that disease. So that is the basic concept behind vaccination. Now there are various places where we can actually go for vaccination. There can be healthcare professionals office, it can be a neighborhood pharmacy, it can be a clinic, community health clinic, it can be a health department, or it can be your workplace also, like schools and educational institutions. Now there are some principles when we are talking about vaccination. What are they? We need to know about the concept of immunity. What is immunity? Immunity means when your body is fighting to give you protection against some disease causing pathogen, or against some component, or against some organism which can cause harm or which can cause a troublesome situation for the normal physiological functioning of the body. And when we are talking about immunity, it is also very important to know the concept of self versus non-self. Okay, what do you mean by self versus non-self? Another important term also comes in connection to this, that is immune tolerance. Okay, so self versus non-self means the ability of our immune system to distinguish between what is self and what is non-self. So self means which is khud ka, apna. isn't it? So self, the body will never ever react, or the body will never ever elicit an immune response against the self tissues or the self cells of the body. Means, say for example, the body will not go and produce antibody against our own heart, or against our own lungs, or against our own stomach. But there are situations when the body actually goes and produces antibodies against self tissues and cells, and those conditions are known as autoimmune disorders or autoimmune diseases. Okay, so that is an exception. That is a type of a disease or a pathogenic condition. But normally, in a normal healthy individual, the cells, or the means the immune cells, will never go and produce antibodies against the self tissues or the self cells. So that is basically the ability of the immune system to distinguish between self and non-self. Non-self means foreign, which is coming from outside. So anything which will be coming from outside, the body will treat it as a foreign element and will start producing an immune response. But there an important factor will come in, that is something called antigen specificity. Means what? The immunogenicity or the antigenicity means the foreign particle which is coming inside our body, that particle needs to have a immunogenicity or a antigenicity. Then only the cells of our body, means the immune cells of our body, will be triggered. Okay, so if there is a foreign particle, or if there is a foreign pathogen which is not having any immunogenicity or antigenicity of its own, then that means the body will not be able to produce any kind of, or the body will not actually go and produce any kind of antibody or immune response against that particular particle, because it is not having any immunogenicity or antigenicity. Okay, so that is there. And it is in and antigen specificity is also very important. Okay, because antigen antibody reaction is something which is very specific. Okay, so against a particular, or against a specific type of antigen, the body will produce only one specific type of antibody, and if that antigen changes, then the antibody will also change. That antibody will no longer be able to go and uh, what to say, bind or kill that particular antigen if it changes its specificity. Okay, so that is again something important. Moving forward, effectiveness of vaccinations. So the small percentage of recipients will respond poorly, which is the role of genetic determinants, because we, each one of us, are different from each other on the basis of genes, on the basis of our genetic makeup. Okay, so it's not necessary that if we are vaccinating, say for example, we are giving COVID vaccination to 100 people. So that's, for example, the same vaccine, Covaxin only. Okay, we are giving Covaxin only to 100 people out there. So we are trying to immunize or vaccinize these 100 people against COVID virus, COVID-19 virus, using the same vaccine. Okay, but it doesn't mean that all of those 100% will respond to the same level. Okay, means the immune system of all of those 100 recipients will be active, like actively participating in the same manner. No, never. Some will respond in a good manner, some will respond in a poor manner. So that will depend on the genetic determinant or the genetic variance. Okay. Now, something called herd immunity, very important when we are talking about vaccination, particularly which was seen in case of COVID pandemic, towards the end of the COVID pandemic, because most of the population had reached herd immunity by that time. Okay, so what is it? Majority of population is immune, so chance of susceptible individual contacting infected individual is low, which is all, which was majorly seen in case of measles epidemic, and nowadays it is also, we can also include COVID pandemic as an example of herd immunity. Okay, measles is a type of a disease which is caused by the measles virus. Okay, so it is a very common type of a disease, and it is not that much threatening also. It mostly occurs among the children. It is a common cough and cold type of a situation. Okay, and small boils and rashes also occur in the body. Okay, so it is not something to panic about, but again it's a very contagious disease. Okay, it easily spreads from person to person. So herd immunity is a term which is used in connection to those diseases which are contagious or communicative diseases, infectious diseases. Okay, say for example COVID-19. So basically what happened after like herd immunity is when around 70 to 80% of the population is already immune. Means either they have been vaccinated, immunized, or they have already contracted the disease multiple times, so now they have automatically got immunized. Means natural immunity or passive immunity, in either ways they have been immunized. So what will happen, the remaining 25 or 30% of the population, maybe they had not yet been immunized, or vaccinated, but still the risk of this person getting contaminated is already lowered, automatically lowered. Why? Because most of the people are immune. So the chances of the infection spreading from the infected person to the uninfected person or the susceptible individual has been increased, has been decreased, isn't it? Because majority of the population is already immune. So they will no longer be going to be infected. So automatically, the remaining smaller proportion of the population that is left behind, they are automatically being protected from the disease. So that is the concept of herd immunity, which is very important to understand when we are talking about vaccinations. Okay. Okay, so I have just kept a slide about herd immunity. So there are several factors which will be affecting herd immunity. What are they? Environmental factors. Okay, means crowded conditions, seasonal variations. Because depending on the season also, there are many diseases that we contract, especially during the seasonal change. Mostly the cough and cold, the flu that we have, influenza, para-influenza. Okay, measles, this type of conditions. Then we have chicken pox also. These are some of the diseases which occur during seasonal change, during the spring and autumn seasons mostly. Okay. Strength of individual's immune system. Okay, because again, as I mentioned, we are all distinct from each other, isn't it? So the extent to which I will react to a particular disease or my body will react to a particular vaccine will not be the same as your body will react. Okay, always there will be some kind of difference. Infectiousness of disease means how much fast the disease is spreading, how much contagious the disease is. Greater the risk of infection, higher percentage of people will need vaccines to attain the herd immunity. When enough people are vaccinated, chance of germ infecting the non-immunized population is small, exactly the concept of herd immunity. And it can also lead to disappearance of disease which actually occurred in case of smallpox. Smallpox is again a disease which is eradicated from the world, like yellow fever. No longer smallpox like occurs, or no longer any human contracts smallpox nowadays. Okay, so obviously if a disease is eradicated, then obviously no vaccination is also needed for that disease. Okay, so this is basically a graph which is showing some like it's giving a kind of a proof about the effectiveness of vaccines. Okay, so we have rubella, which was introduced in the year 1969. Then we have paralytic polio vaccine, 0 cases. Okay, so these are basically in the x-axis we are showing the number of reported cases, and on the y-axis we have the different diseases. Okay, so we have pertussis, then we have mumps. Okay, so see, dhire dhire the occurrence is actually decreasing after the vaccination. Okay, diphtheria is almost gone, measles have been halved, mumps has also been halved, pertussis also almost halved, paralytic polio is gone. Okay, rubella is also almost halved. Okay. Okay, now coming to development of vaccines. Okay, so there is some common misconception that activation of the immune system results in protective immunity. Multiple factors are there which affects decisions when when we are making when we are going to come up with a new vaccine. What are they? Number one, activation of specific branch of immune system means which part of the immune system or which branch of the immune system we are actually targeting, whether cell mediated immunity or whether the humoral mediated immunity. Okay, and also development of immunological memory, because development of memory cells is the most important part of vaccination. Okay, if as a result of vaccination the memory cells are not formed inside the body, then the vaccination will be of no use, because when the person is actually going to contract the disease, if the memory cells are not there to remind the immune system that okay, you have antibodies against this pathogen already, then that means the vaccination has failed, isn't it? So these are the two most important aspects when we are talking about developing a vaccine. Okay, now coming to the role of memory cells, very important. So it will depend on the incubation period of the pathogen, means how much time the pathogen is actually incubating inside the body to cause the disease. Okay, so if the incubation period, there are some pathogens for which the incubation period will be short. Say for example influenza, which is causing common cold. So the symptoms already underway by the time the memory cells are activated. Repeated immunization with neutralizing antibodies. So basically, if the if the incubation period is short, then what will happen? By the time the memory cells are activated, because to become activated, the memory cells will need some point of time, some amount of time the immune system will need in order to go and activate the memory cells. So if the incubation period is short, then what will happen? Before the memory cells could be activated, the job will be done by the pathogen. Okay, the person will already start suffering from the disease before the memory cells could even be activated. So in those cases, we need to give frequent booster doses. Okay, but if the incubation period is long, say for example in case of poliovirus, the incubation period is long in comparison to influenza. In such case, the body is having enough time to allow the memory cells, particularly the memory B cells, mean the B lymphocytes, because they are involved in production of antibodies, to respond. Okay, so by the time the disease actually develops, the body will be getting enough amount of time to go and activate the memory cells that, okay, the pathogen has come, now we need to produce the antibodies. Okay. So again, I've given a graph. Okay, on the x-axis we have the time on the months, and on the y-axis we have the mean antibody titer, means the level or the load of the antibody that is present inside the body with respect to the time. Okay, so see, more the time, the immunologic memory is going to be at a point of after a point of time, the immunologic memory is reaching a stationary phase. Okay, so see, it is increasing, I think, till around 3 months. Around 3 months time, the immunologic memory is increasing, but after that gradually it is reaching a stationary phase, means it's no longer increasing nor decreasing, it's the same, it's remaining the same. And talking about the serum antibody level again increases shortly after vaccination. Okay, before 1 month, before the span of 1 month only, jitna usko upar jana hai, the like uh, the maximum level is attained before 1 month only of the vaccine. Okay, sorry, not of the vaccine, of the serum antibody which will be produced in response to the vaccine. After that, slowly the level of the serum antibody is getting decreased as the time is increasing, as the month is increasing. See, up to the 12th month the level has already come down. Okay, so that means that is where we can explain the need or the necessity of booster dose to boost the memory cells. Okay. Okay, now coming to the types of vaccines, very important. So first we have the whole organism. Whole organism means where the entire microorganism which is responsible to causing the disease, the entire pathogen which is responsible to causing the disease is used to prepare the vaccine. So whole organism mein we have two types. One is activated, activated also, or we do not usually use the term activated. We use the term attenuated. Okay. So attenuated means something whose attenuation has occurred. Okay. Attenuation has occurred means only the infective part of that material has been taken out. Okay, only the part which is responsible to cause the infection is taken out. The remaining part of the organism is intact. So that means it is attenuated. Only the infectivity has been lost, has been made to lost rather. Okay. And another one is inactivated, means it's killed. The virus or the bacteria or the fungi, whatever it is killed. That means inactivated. Then we have purified macromolecules. So it will contain the different, like the capsid or the toxoid. Toxoid is particularly used in terms of bacteria. Okay. Recombinant vector, recombinant antigen, polysaccharide, means particular parts of the infective material is being used. Okay, so those parts are isolated and they are being purified. So that will come under the category of purified macromolecules. Then we have DNA vaccines, we have synthetic peptide vaccines, and we also have multivalent subunit vaccines. We also have recombinant DNA vaccines, which will also come under DNA vaccines. Okay. So this is basically a chart that I have given which contains the different whole organism vaccines that we have for humans. Okay, so under the whole organisms, among the bacterial cells, we have anthrax, which is inactivated. We have cholera, which is also inactivated. Pertussis inactivated. Plague inactivated. Only tuberculosis and typhoid will be having the live attenuated virus. Then talking about the viral particles having whole organism as vaccine, we have hepatitis A inactivated, we have influenza inactivated, then we have measles which is live attenuated, polio. Now in case of polio, we have two types of vaccine. One is Sabin and one is Salk. Okay, so Sabin one is the live attenuated vaccine, and the Salk's vaccine or the Salk vaccine is the inactivated or the killed vaccine. Okay, then we have rabies, which is again live attenuated. Then we have rubella, which is inactivated. We have varicella zoster virus, that is chickenpox virus, which is live attenuated. Then we have yellow fever vaccine, which is also live attenuated. And then talking about the purified macromolecular vaccines, we have toxoids. Toxoids mein sabse important is diphtheria, which is an inactivated exotoxin. Then we have tetanus, which is again inactivated exotoxin. Then we have capsular polysaccharides like Haemophilus influenza, which is having the polysaccharide as well as a protein carrier with it. That is Haemophilus influenza type B, because in Haemophilus influenza have various types. Okay. Then we have Neisseria meningitis, which is containing the polysaccharide. Then we have Streptococcus pneumonia, which is having 2 to 3 distinct capsular polysaccharides. And then we have surface antigen, under which we have hepatitis B virus vaccine, which is having a recombinant surface antigen, that is, which is abbreviated as HBs antigen. Okay. Okay, now talking about the attenuated viral or bacterial vaccine. So attenuation means, what is attenuation exactly? To reduce in force, value, amount or degree, basically to weaken. So by removing the infective part, we are actually weakening the pathogen. Okay. So usually the attenuated vaccines that we have, they are low risk vaccines because there is no risk of reversion and causing the disease, but they need booster doses because it is the inactivated form of the pathogen. Okay, so the immune cells will be elicited, the immune response will be elicited, but the degree or the extent to which the immune system will be triggered will be less, as a result of which booster doses will be required. That is the basic difference between attenuated vaccine and inactivated or killed vaccine. Okay, so they usually achieve by growth under abnormal cell conditions. Ideal example will be BCG, Bacillus calmette-guerin, which is the first vaccine that is given right after birth, vaccine against tuberculosis. It acts as a double-edged sword, as they have distinct advantages and disadvantages. I think I already talked about the advantages and disadvantages, but let's see. Talking about the advantages, advantages stem from their capacity for transient growth, prolonged immune system exposure, single immunization, and replication within the host cells. Because it's activated, because it's not killed, so single dose will be enough, no booster dose will be required, as I mentioned, and it will be replicating within the host cells. Okay. Now there are some exceptions to the advantages that we saw of the attenuated vaccines. What are they? It will come in the case of polio virus. So as I mentioned, polio vaccine can be of two types. Sabin will be the attenuated or the live virus vaccine, and Salk will be the dead or the killed or the inactivated vaccine. Okay. So here, since we are talking about the attenuated or the live vaccine, so here we will be focusing on the Sabin polio vaccine. So it consists of 3 different attenuated strains of polio virus. Colonization of intestine will result in immunity to all the 3 strains, and it will be producing secretory IgA, and it will be also inducing production of IgM and IgG. And the result will be the need for the boosters. So in spite of being a attenuated viral vaccine or a live viral vaccine, it will be requiring booster doses, which is an exception to the features of to the typical features of the attenuated vaccines. Okay, so individual strains will interfere with one another. Why? Because there are different strains. Okay, so if we are giving a vaccine against one strain, then the other strains might be requiring an effective immune response again. So that's why it will elicit the need of a booster dose, of a second dose. So the after the first immunization, one strain will be predominating in growth. After the second immunization, immunity will be generated by previous immunization, and it will be limiting the growth of the previously predominant strain, but not the second predominant strain. And that's why we will need a third immunization also, which will be working in the same principle with the second immunization. So after 3 rounds of immunization, we can say, okay, now that person is safe from the polio virus, because it is having 3 different strains. Okay. Now coming to the disadvantages, major disadvantage will be possible chances of reversion. As I mentioned, since it's a live pathogen, so it can happen that instead of giving protection, it is actually leading to the disease inside the body. Okay, and it was actually found in case of polio vaccination. It was seen in around one case in about 4 million doses. It's a very unfortunate incident, but it did occur. Okay, so there is a remote possibility that the person may actually suffer from the disease after taking the vaccine, if it's the Sabin vaccine, if it's the live vaccine of the polio virus. Presence of other viruses as contaminants, very important, again for the same reason, because it's a live virus, or it's a live pathogen, isn't it? So if that pathogen is also having some other virus or other bacteria inside its body, then that will also be going to get introduced inside the body of the patient, isn't it, or the recipient. And also unforeseen post-vaccine complications, it can lead to some post-vaccine complications which are unwanted or unexpected. Okay, so I think that's it. In the second session of this topic, we will be talking about the other types of vaccines, and majorly I want to talk in details about COVID vaccines, because COVID pandemic is something that we all of us have faced, and it is something that each and every one of us must be knowing at least in some details, and particularly those people who are coming from medical science background, like you people who are taking a course on medical biotechnology. So it's very important that you know the basics and the important aspects about COVID-19 virus and also COVID-19 vaccination. Okay, so we will be talking about that in the next upcoming session. Till then, thank you, goodbye. Please like, share and subscribe ITLS Academy. Thank you.